Hello everyone, and welcome to our last lecture of unit number 1. You have made it this far, congratulations on that, and you're almost done with the unit. So in this lecture, we will be talking about the structure and function of acellular microbes. As always, you have your lesson objectives here. Hopefully by now you guys know how to use them in the most effective way, but kind of the way I recommend doing it is look over them now before you start the lecture so you know what you're listening for, and then look over them again at the end to make sure you got all the key points of the lecture. If you can think back to our first lecture about the different types of microbes, I mentioned that there are two types of acellular microbes. There are the viruses and the prions. Viruses are composed of protein and nucleic acid, and prions are just composed of protein. For the first part of this lecture, we will be focused on the viruses, and then for the last couple of slides, we'll talk about the prions. Viruses and prions were some of the last microbes that were discovered. Viruses were discovered basically because scientists were realizing that while things like bacteria and viruses and fungus could infect organisms and cause disease, in some cases when they would study the diseases that were caused or that were showing up in things like plants and animals, they realized that there must be something smaller than a bacterium that was causing disease, because they would look at the samples under the microscope and they would find nothing. And so they realized that there was something smaller than a bacterium that was causing disease. And eventually people were able to start to visualize these particles that scientists had figured out existed but couldn't see just yet. With the advent of the electron microscope, people were able to see that there were numerous different types of viruses that existed in the world, and they were all incredibly small. So just for reference, so you understand how small we're talking about in terms of the size of viruses, this is, or at least on the slide, this represents a human liver cell, which, as you know, is very small in itself. You would not be able to see it if you were just presented with one on a slide. Right here is a bacterium. It's the E. coli bacterium, which is a relatively small bacteria compared to other types of bacteria. And then within that type of bacterium, we have the viruses. Viruses themselves range in size as well. So sometimes we have incredibly small viruses, we have some that are a little bit larger, and you can see they have different shapes to them in addition to their different sizes. So we're going to talk about the different shapes, some of the different types of viruses in this lecture, but I just wanted to give you a visual of and a comparison of viruses to other types of microbes and other types of cells. The outermost layer of a virus is basically its outer covering, or its shell, is called a capsid. And the capsid is composed of proteins. When you join a bunch of individual subunits together, that's when you get the capsid. And the individual proteins that join together to make the capsid are called capsomeres. So in this top picture up here, all of these individual they're kind of oval shaped, more oblong shaped, but all of these individual units are proteins that are joined together. And when you join those proteins together in the specific pattern that's necessary, they will form the capsid. So each one of these individual little guys is a capsomere. And when we join enough of them together in the right pattern, that is a capsid. Capsids come in two main shapes. There are exceptions to this, just like there are exceptions to virtually everything in life. But the two main shapes of capsids are helical, which is rod-shaped, and that would be the one up here, and icosahedral, which looks round when you put it under a microscope, but it's actually a kind of a sharp-sided figure, oftentimes 20-sided figure, that is composed of kind of individual subunits of triangles that all get put together. So just to make sure these are labeled appropriately for you, this is the helical capsid, and this is the icosahedral capsid. In terms of the properties of capsids, Capsids themselves are fairly resistant to destruction. They're not as good as something like an endospore, and may not even be as good as something like a protozoal cyst. However, they're pretty sturdy little structures, and they can be difficult to clean off surfaces or clean off objects. So oftentimes when we know we're working with some kind of virus, we need to be aware of the fact that capsids can be difficult to destroy, and as a result take extra precautions or use a special type of disinfectant to deal with that. Now, in many viruses, the capsid is the outermost layer of the virus. It's just the outer structure of the virus, and that's it. There's nothing more on the outside. These viruses are called naked viruses. So viruses that just have a capsid and nucleic acid on the inside are called naked viruses. But some viruses have an extra covering on the outside called an envelope. The envelope is made up of membrane from a host cell. That could be the plasma membrane, that could be the nuclear membrane, if we're talking about a eukaryotic cell, that could be the membrane from the endoplasmic reticulum or the Golgi apparatus. There are different locations that a virus could get a membrane from. Within this membrane are things called spikes. So these structures sticking out of the membrane are called spikes. The membrane comes from the host cell or the cell that the virus was living inside. But the spikes are actually made of viral proteins. So on this diagram here, we have the capsid, which in this case is the icosahedral form. We have the nucleic acid, which is inside the capsid, and we'll talk about the nucleic acid more shortly. But for now, we've got the nucleic acid inside the icosahedral capsid. And then we have this membrane covering that's called the envelope. This envelope is obtained from the host cell of the virus, and the host cell is what the virus is living inside. And the membrane portion of the envelope comes from that host cell. But these spikes on the outside here come from the virus itself. The importance of the spikes are that they are used by this virus to attach to the next host cell that it wants to invade. So the envelope of some viruses is significant in that, one, it has these viral proteins that allow it to attach to the next cell that it wants to invade. And two, it has this extra covering over the capsid that helps protect it from things like the immune system. It kind of acts as like a shield or a cover or a way to disguise the virus from the immune system, so the immune system can't find it quite as easily. So now that we've talked about capsids and the two main varieties there are, as well as the naked viruses versus the enveloped viruses, we'll talk a little bit about the genetic material. Viruses are interesting in the microbial world because microbes that are composed of cells, things like bacteria, protozoa, helminths, algae, etc., fungi, they all have DNA and RNA. Viruses, though, can have DNA or RNA. They don't have both. So a virus will either have DNA or it will have RNA. And another unique thing about that is that this genetic material can be either double-stranded or single-stranded. Typically, DNA is double-stranded, but in viruses it can be single-stranded. And typically, RNA is single-stranded, but in viruses it can be double-stranded. So there are kind of like four main types of genetic material or nucleic acids that a virus could have. It could have double-stranded DNA, it could have single-stranded DNA, it could have double-stranded RNA, or it could have single-stranded RNA. So those are kind of the four main combinations or types of genetic materials that a virus could have. Now within this genetic material, there are genes, and genes are sequences of nucleic acids that code for proteins. Some viruses have a lot of genes, some of viruses have not very many genes. And one thing that's important to keep in mind is that the number of genes viral DNA or RNA codes for is not directly related to the pathogenicity of the virus, which means the ability of the virus to cause disease or the severity of the disease that is caused. So just as an example, human immunodeficiency virus, the virus that causes HIV, has 9 genes, and herpes has 100 or sometimes more genes in it. So HIV is a potentially lethal infection, where herpes is almost never a lethal infection. Obviously, the 9 genes that HIV has can cause a lot more damage to the human body than the 100 genes that the herpes virus has. So far, we've talked about the structure of viruses. We've talked about their outer covering, which is called the capsid, and how some of them have an extra layer called the envelope. We've also talked about the types of genetic material that could be inside the capsid of a virus. And now it's time to switch gears a little bit and talk about viral lifestyle. Viruses require a living host to be active. Viruses are what we call obligate intracellular parasites, which means obligate means they have to do something. Intracellular means within cells, and parasites means they have to be within a living cell. So a parasite is something that lives on or inside a living being. So viruses themselves cannot function outside of living cells. They can exist outside of living cells, but when they exist outside of a living cell, when they are on a tabletop or a pen or a doorknob or are floating in the air because someone just sneezed them out, they are basically inert. They're not doing anything. They only become active when they're inside a living cell. The main job or function of a virus is to infect host cells, which means invade a living cell, and replicate or make lots of copies of themselves. That is what a virus is programmed to do. All of the genes that are contained in the genetic material or the nucleic acid of a virus, whether that is DNA or RNA, are there to help the virus create proteins that will lead to more viruses being made. And then once those new viruses are made, the goal of those new viruses is to leave their current host cell and go to another host cell so they can use it to make copies of themselves. Typically, viral replication damages the cells that are being used as hosts. So the cells that are infected become damaged as a result of the viruses being inside them and using their cellular components to make new copies of the virus. The damage that is caused by viruses to cells is called a cytopathic effect. And cytopathic effects occur because the virus that is inside the cell, using the components of the cell to make copies of itself, interfere with the metabolism and the replication of that cell itself. So if a cell is infected with a virus, it is not able to metabolize as effectively as it normally can. It may not be able to reproduce as effectively as it normally can, or it may be able to reproduce, but not in the normal way that it should be able to reproduce. And in some cases, these viruses, when they're inside individual cells, actually just destroy the cell itself and cause it to burst open or just stop functioning and cause it to die. We can see cytopathic effects both in a microscope and in the body in some cases. So what we have here, the little pink round looking things, are viruses. We have some cells that have been infected with the virus. And you can see we have one cell here that seems to have not been infected yet, but we have a bunch of broken open cells here with a bunch of cellular debris surrounding as well. So what this picture is showing us is that when these viruses get into these cells, it causes these cells to break open and spew their contents into the external environment. And those are the cytopathic effects that we see under the microscope. This person has a cold sore, which is caused by a herpes virus. And we can see the cytopathic effects of that virus just on her body. At a cellular level, this is probably similar to what's happening. And that at a macroscopic level, we can see the cytopathic effects because we can see that the cells around her lip have essentially broken open. They've burst and they've caused this open sore to form on her skin. And that's another example of the cytopathic effect just at a macroscopic level that we can see. Now, we've established that all viruses have to be inside a living host cell to become active. But there are different types of infections that occur depending on what specific type of virus we're talking about. So in terms of the three main type of infections that occur, we have acute infections, latent infections, and persistent or chronic infections. In acute infections, we have an immediate infection. Once the virus enters the body or enters the host cell, it starts to cause damage right away, make a bunch of copies of itself, and it causes immediate results. Those results may not show up for a day or two until the virus basically gathers in large enough numbers to cause noticeable effects. But in viral terms, that's immediate. So it causes results right away. And then once the virus runs its course and the immune system takes care of it, the virus is cleared from the body. It is no longer there. So when we look at that type of infection on a graph, if we have time on the x-axis and viral load on the y-axis, that type of infection would look like this. We go from zero virus to a lot of virus and a lot of effects, back down to zero virus. And then the virus is gone again. So this would be an example of how an acute infection would look. If we were mapping time against viral load and effects of the virus. So here we start at 0.0 when the person just gets infected with it. There's not very much virus in the body or no virus in the body. The person gets infected. The number of viruses or the viral load rises. It peaks, and then the immune system takes care of it. It starts to drop and goes down to 0. This is when we would start to see like the worst of the signs and symptoms for the most part. Then those signs and symptoms would decline as the level of viruses in the body drops as well. Examples of acute infections are things like colds and flu and certain other respiratory viruses or sometimes intestinal viruses where you have a definite starting point and a definite ending point. With latent infections, they mimic acute infections, at least initially, but the difference between those is that with these types of infections, the virus is not actually cleared from the body. It stays in the body in a dormant form and then, depending on various environmental conditions, becomes reactivated again. So a latent infection would look like this. You would start out at time 0 where the person just becomes infected. The number of viruses would rise, would peak, and then would fall, but never really gets back to 0 because it's going to stay in that person's body. Then we're going to keep really low levels of the virus until something in the environment causes it to rise and then fall again. And then it's going to go on until it rises and falls again. And basically, good examples of this type of infection are things like the herpes viruses. So we're just talking about cold sores on the previous page. When someone gets a cold sore, basically what has happened is they have come in contact with a virus, a certain type of herpes virus, and they start out with 0 viruses in the body, but they get infected by sharing a drink or kissing or some other kind of activity where some type of saliva is shared. And the number of viruses rises, they get their initial cold sore outbreak, and then the number of viruses starts to drop, the cold sore goes away, but it doesn't ever fully get cleared from the body. So that virus is just going to hang out in the body, and then something in the environment is going to trigger it to become reactivated. So it will become reactivated. It's going to create a bunch of copies of itself, so the viral load or the number of viruses will rise. It will peak, the immune system will take care of it, it will fall again, and then stay at a fairly low level until again something in the environment causes another outbreak to occur. A persistent or chronic infection, these words are basically interchangeable, causes immediate infection and immediate results, but the infection doesn't have this cyclical nature the way a latent infection does. Oh, so let me write latent up here. Instead, the viral load continues to rise constantly. So we don't have this cyclical nature of rise and fall and rise and fall. It rises constantly, and sometimes it can rise slowly, sometimes it can rise fairly quickly. But in a persistent or chronic infection, basically we start out at the time of infection with 0 viruses. It will rise to a peak, and then usually it'll drop off slightly as the immune system deals with it a little bit. But after that, it'll reach kind of its low point, and from there, it will just keep climbing over time. So a good example of this type of infection would be HIV, especially if it's untreated. So when someone gets the human immunodeficiency virus, they start out with 0 viruses in their body. Then they get infected. The number of viruses rise. They have some signs and symptoms. And then oftentimes the immune system will take care of kind of that initial infection, get rid of it, make the signs and symptoms go away. That'll drop off. And then just slowly over time, the viral load or the number of viruses will continue to climb. This is if it's untreated. If a person is able to get treatment and uses that treatment consistently, they can keep their level of viruses very low. But let's say just in an untreated HIV infection, this is what the graph would look like. Now, some viruses only will cause acute infections. Some will only cause latent. Some will only cause persistent or chronic infections. But there are a few out there that could cause basically either acute or persistent, or sometimes either acute or latent. But most of the viruses fall into just being able to cause one of these types. For example, a cold virus, let's say you just get a typical standard cold. We're not talking about COVID. We're not talking about anything else, just that your typical standard cold can only cause an acute infection. It can't cause a latent infection because it doesn't stay in the body. And it can't really cause a persistent infection because our immune systems are developed to the point that they just get rid of that virus. So basically, most viruses fall into just one of these categories. Occasionally, we have a virus that can kind of switch lifestyles on us, but most of the time, if you have virus A, it's always going to cause an active infection. Virus B will always cause a latent infection. Virus C always causes a persistent infection. They don't usually switch it up very often. Now, there is one more type of viral infection that I didn't include on the previous slide because it's basically a subset of persistent infection. And this type of virus, the type of virus that causes this situation, is called an oncovirus. Oncoviruses are viruses that can lead to cancer. Oncovirus infection doesn't always lead to cancer, but we know for sure that there are some viruses that when they infect people, they can lead to persistent infections that will cause cancer. In cases where these oncoviruses are causing cancer in people, what the virus does is it causes the host cell to become transformed. And what that means is this virus, when it infects a cell, it causes a cell to lose its normal characteristics and lose its ability to perform the functions that it's supposed to do. So basically, the cell loses its purpose And in addition to losing its purpose and losing its ability to function normally, it also starts to divide rapidly. So now, instead of having, we'll just take a liver cell, for example, instead of having a standard liver cell that is doing its job and detoxifying things that pass near it, the liver cell loses its ability to detoxify things. And instead of doing its normal job, it just starts to replicate really, really rapidly. What this causes, or what this can lead to, is the growth of tumors. So tumors are essentially a mass of cells that have grown as a result of some type of cellular transformation. That transformation can occur for lots of different reasons, but there are some cancers, some types of cancers, and some types of tumors that arise as a result of oncoviruses. Some examples here are human papillomaviruses. There are actually lots of different types of human papillomaviruses, but certain types of them can lead to certain types of cancers in people. So cervical cancer, certain types of oral cancers, other types of genital cancers, those can all be caused by certain types of the human papillomavirus. The Epstein-Barr virus, which is the virus that is typically associated with mononucleosis or mono, can also lead to cancer in some cases. It's not very common in developed nations, but in people who are chronically infected with this, or basically get repeated infections of this, and also have other health conditions, especially malaria, it is much more common for this virus to basically become oncogenic and cause cancer to form. And then hepatitis B and C viruses can also become oncogenic, meaning they can cause cancer. They don't always cause cancer, but they can lead to cancer formation in some people. Now that we've talked about the structure of viruses and the types of diseases they cause in terms of latent disease, persistent disease, acute disease, cancer, etc., it's time to talk about how viruses actually invade cells and how this leads to cytopathic effects, or how this leads to cell damage. Now, there are lots of different types of viruses in the world. There are viruses that infect people, there are viruses that infect animals, that infect plants, that infect bacteria, that infect fungi, etc. Basically, as far as we know, every type of cell on the planet has at least one virus that infects it. Now, viruses tend to be very specific in terms of what they infect. So most viruses can only infect one to a few types of cells. This is why, even though there are many, many different cell types out there, and as far as we know, every cell type has at least one virus that infects it, it doesn't mean that we're constantly getting viral infections, right? Because viruses are very specific in terms of which cells that they will actually infect. So even though we're surrounded by viruses and viruses infect everything, we're not constantly getting viral infections. On this slide, we are just going to talk about animal virus replication. There are, like I said, lots of different types of viruses out there, and plant viruses replicate a little bit differently than animal viruses. They replicate a little bit differently than bacterial viruses and fungal viruses, etc. So we are going to focus, at least on this slide, on animal virus replication, because we are animals, and when we're talking about people going into the healthcare professions, their main focus and the main types of viral infections they will be dealing with are animal viruses, because people are animal viruses. So we'll talk about how animal viruses replicate themselves and cause disease, or cause those cytopathic effects. There are 5 stages or 5 steps to animal virus replication. The first step is what's called adsorption, and that is with a D, not with a B. It's not a typo there. Adsorption is the process of a virus sticking on to the outside of a cell. So the virus sticks to the host cell on a specific receptor. Typically, The receptor is a piece of protein that's in the plasma membrane of the host cell. It doesn't have to be, just typically it is. And the match between the virus and the receptor has to be exact. This is the reason viruses can't just infect every cell type. The ability of the virus to actually stick on to that protein has to be basically a perfect match. And if that perfect match doesn't occur, then the virus is not able to infect the cell. Now, adsorption occurs a little bit differently depending on whether you're talking about a naked virus or an enveloped virus. A naked virus, just part of the capsid, attaches to the receptor, whereas in an enveloped virus, the spike protein will attach to the receptor. Either way, ultimately what's happening is the virus is attaching to a receptor that's located on a host cell, and it's getting ready for the next stage of replication. The next stage is called penetration. Penetration is when the virus is going from the external portion of the cell to the internal portion. So this is basically the virus is making its way through the cell membrane somehow and getting into the cell. In addition to entering the cell, the virus goes through a process called uncoating. So uncoating occurs when the genetic material that's in the capsid is actually freed from that capsid. The capsid might dissolve or it might open up some way, but either way, the genetic material essentially escapes the capsid. Penetration and entry and uncoating happen in slightly different ways, again, depending on whether we're talking about an enveloped virus or a naked virus. Naked viruses typically are brought into a cell through an endocytic vesicle. If you think back to the eukaryotic lecture, that was one of the types of vesicles I mentioned on that slide about vesicles. But basically what happens is with a naked virus, the capsid attaches to a receptor in the cell, and the cell notices that there's something sitting on its surface. So it takes some plasma membrane and it kind of surrounds that virus. It brings it into the cell by using its own plasma membrane. With enveloped viruses, there's kind of a little bit of a different way of happening here. What happens with an enveloped virus is the envelope, which is itself membrane, kind of just attaches itself to the cell membrane of the host cell, and the capsid sort of just sinks through that membrane into the cytoplasm as a result. So in that case, the cell doesn't have to actively bring the virus in. It's just the membrane of the virus, the envelope of the virus, attaches to the membrane of the host cell, and that allows the capsid to be delivered into the cytoplasm. After entry and uncoating occurs, the next stage is synthesis. In synthesis, the genetic material of the virus is used to create viral proteins and to make more copies of the genetic material. So the genetic material is copied. There's a lot of copies made of it. And that genetic material is also used to make the viral proteins that will create the capsid. Once the correct number of genetic copies have been created and enough proteins have been made, the next stage can begin to occur. That next stage is called assembly. And during assembly, the capsid is formed and genetic material is put inside the capsid. So that process of putting the genetic material inside the capsid is called encapsidation. And once assembly is complete, basically once the genetic material has been put inside the capsid, the final stage is release or exit. New viruses exit the cell either by budding out or by bursting. Budding is kind of shown here, where the virus essentially leaves the cell relatively slowly and relatively gently Oftentimes it is surrounded by some kind of membrane. It's in like an exocytic vesicle, or it's in some kind of membrane coated vesicle. And that coated vesicle just bumps up to the cell membrane of the host cell, and the virus is released. Bursting occurs when a bunch of viruses build up inside of the cell, and the cell essentially gets too full. It's too full with viruses, and so it explodes and releases the individual viral particles into the external environment. So oftentimes budding occurs with enveloped viruses and bursting occurs with naked viruses, but not always. That's not a hard and fast rule. One thing that's important to remember is that oftentimes only one or a few individual viruses will go into a cell, but hundreds or thousands will come out of a cell. So even just one virus entering a cell can sometimes be enough to create thousands of more copies of that virus. And so that is why sometimes you start feeling so bad a few days after you've acquired a viral infection, right? You start feeling a little bit sick on day 1. You feel a little bit worse on day 2, but not terrible. And then by day 3, you've got trillions of copies of this virus in you, and they're all invading your cells and causing them to burst open, or causing them at least to bud open and taking maybe envelope with it, so your cell isn't super stable anymore. And so you start to feel really bad after a couple of days because the number of viruses has built up over time, and they're all sitting there doing damage. On our last slide, we talked about animal virus replication. And we focused on that because most of the viruses that healthcare professionals will deal with will be animal viruses. Whether you're going into human healthcare or animal healthcare, either way, you are going to be dealing with viruses that infect animals. And so that's why we focused primarily on that. However, there is another type of virus that is important to future healthcare providers. Things like plant viruses, viruses that infect fungi, viruses that infect archaea, viruses that infect algae, they're not quite as significant as the animal viruses and then the viruses we're going to talk about right now. So there is a type of virus that infects only bacteria, and that type of virus is called a bacteriophage. There are many, many different types of bacteriophages. Some people even think that bacteriophages might be the most common type of biological entity on the planet. Basically, we are surrounded by bacteriophages all the time. However, most of them don't play a big role in our lives because they only infect bacteria. In terms of their structure and function, they are similar to animal viruses, but not entirely identical. If you look at the example of the phage I have over here on the right-hand side, you'll see that its structure is a little bit more complex. We have the top part of virus. Some people might call that the head, and it's basically the icosahedral capsid with the genetic material inside. And then we have a long tube. Many people refer to that as the tail. And then these little spindly things that stick out that look kind of like legs, those are called tail fibers. And so in many ways, bacteriophages behave in a similar way to animal viruses, and they have similar structures. They just are sometimes a little bit more complex in how everything is put together. The reason I am mentioning them now and why healthcare providers at least need to know a little about these types of organisms is that occasionally when a bacteriophage infects a certain type of bacteria, it can make that bacteria more pathogenic. Now, there are a couple examples of this, but one of the examples, kind of the best known example, is a situation with a bacteria that causes a disease called diphtheria. Don't worry if you don't know what diphtheria is right now. You will learn about it in this class, I promise. But basically, this disease called diphtheria is caused by a bacterium called Corynebacterium diphtheriae. And that bacteria actually is generally non-pathogenic, meaning most of the time it doesn't cause disease. In fact, many of us can carry that bacteria in our mouth, around our mouth, kind of on our skin in general. Many people will have it in the throat, various regions on their body, typically in the head region, but it could be really anywhere, I suppose. And most of the time, it doesn't cause disease. However, if that bacterium, Corynebacterium diphtheriae, gets infected with a particular bacteriophage, then what it does is start producing a toxin. And that toxin causes disease in humans. So this bacteria that causes this disease is relatively benign until that bacteria itself becomes infected with a bacteriophage. And then kind of the response of the bacteria to that infection is to produce this toxin that ends up causing damage in the human body. So in that regard, we do care about bacteriophages because although they don't necessarily infect our cells directly, they can make us sick if they infect bacteria that happen to be on or inside our bodies, and the bacteria then become pathogenic as a result. Another interesting thing about bacteriophages is that sometimes they can be used to treat bacterial infections. It is not super common for this to happen in the United States, although it is kind of in experimental trials and there are some situations where it's used. But basically this process of using bacteriophages to treat bacterial infections is called phage therapy. What we can do, because these viruses are not infective to human cells, is figure out which bacteriophage would be useful at killing off the bacteria that a person is infected with. And then from there, we create some kind of solution containing the bacteriophages and deliver that to the person. The idea is those bacteriophages then will go on to infect the problematic bacteria. They will kill off those problematic bacteria and not damage any of the other bacteria that's in the human's body. And that way the person will be able to be treated with something other than antibiotics. So like I said, this is not something super common that happens in the United States. It happens every once in a while, and there are some clinical trials going on with this type of treatment these days periodically. It's more common in other countries. Countries in Europe do it a little bit more often than the United States. But with the rise in the number of antibiotic-resistant bacteria that are out there and the loss in effectiveness of our antibiotics, we may start to see this as an option in more cases. There are two main ways that bacteriophages can replicate. The first type of replication is called the lytic cycle. In the lytic cycle, essentially a bacteriophage injects its DNA or its genetic material into a bacterial cell. That genetic material is copied and viral proteins are produced, and it's very similar in several of the steps to the way animal viruses reproduce as well. So the genetic material enters the cell, the genetic material is copied, it's also used to make viral proteins, the body of the bacteriophage is put together, and then the bacteriophages, the completed ones, the new ones, will burst out of the cell and go on to infect other nearby bacteria. There's another way that bacteriophages can replicate though, and this is called the lysogenic cycle. In this situation, what happens is the bacteriophage injects its DNA into the bacterial cell, and instead of that bacterial DNA being immediately used to make new viral particles, it actually is incorporated into the bacteria's genome or into the bacteria's chromosome. So you can see here we're looking at a situation where the virus we're starting in step one. Step one is the same whether you're talking about the lytic cycle or like a lysogenic cycle, but the genetic material of the virus has entered the bacterial cell, and instead of being used to make new viral particles right away, it actually is going to get added to the chromosome of the bacteria. So you can see the bacterial chromosome in blue, and now it has this new segment in red, which is the viral genome. or the viral genetic material. Either word in that case is fine. What's going to happen now is the bacteria is going to go through normal cell reproduction, and every time it reproduces itself, it's going to copy its chromosome. Instead of just copying its own chromosome, though, it's also going to copy the viral chromosome. So its offspring are going to have that viral DNA inserted into the bacterial chromosome. Now, eventually, some kind of environmental signal will occur, some kind of trigger, that will cause the viral DNA to leave the bacterial chromosome. And at that point, the viral DNA will be used to make new viral particles. And those new viral particles will get put together, they'll get created, and they will end up bursting out of the cell. One of the reasons I like to talk about this at this point in the lecture is not only is it just important to know at least a little bit about what bacteriophages are and their replication cycles, because as we know from the previous slide and the previous information covered there, sometimes bacteriophages can be problematic for people and animals in some cases, but also this lysogenic cycle where the viral DNA gets incorporated into the bacterial chromosome is significant in biotechnology. We can actually take advantage of this type of situation, the ability of viral DNA to be incorporated into the bacterial chromosome, so that we can, if we want to, manipulate the code in that DNA that gets put into the bacterial chromosome and use that to help create certain proteins that we want created. I'm going to talk about that more on the next page, but I just wanted to kind of explain to you why this is important to talk about and give you a little bit of context about why we care about bacteriophage replication. Many times when people hear the word virus, they assume all kinds of negative things. And it is true that in a lot of contexts, in a lot of cases, viruses are not super helpful to humans or to animals or to plants, etc. They can cause sometimes fatal diseases. They can result in the loss of all kinds of money in the economy and just generally cause various problems in society, as you guys are experiencing right now with COVID. However, viruses are also very useful to us in biotechnology and medicine. We can use viruses, and oftentimes with the help of bacteria as well, to create all kinds of products. We can use them to help create insulin, we can create growth hormones, we can create vaccines, cancer treatments, genetic therapy, like I mentioned before, treat bacterial infections with them, and we can even manipulate plants into the way we might want them to have a certain kind of physical characteristic or have them look a certain way. So just keep in mind that just because viruses can be very negative doesn't mean that we can't use them in positive ways to help people in medical settings, to use them in industrial settings if we want to create a certain something. When we're looking this example of the plant, this is just a fun thing to look at. It's not necessarily useful other than it's just nice to look at. But we can take viruses and we can alter plant characteristics to make them more productive or make them more useful in various types of settings. So keep in mind that while viruses have their drawbacks for sure, they are very useful in laboratory settings and in biotechnology settings to help us create medicines and hopefully make the world a better place. The last type of microbe we talk about is a prion. Some people pronounce this prion, and either way is fine. So however you want to say it, however someone else says it, as long as it's prion or prion, that's fine, either way. And what this term stands for basically is proteinaceous infectious particles. Prions are just made of protein. They have no lipids, they have no carbohydrates, they have no nucleic acids. They are nothing but pieces of protein. And one of the especially intriguing things about them is that they are basically just altered versions of normal cellular proteins. So if you want to think of just some kind of generic protein as version 1, then a prion is version 1.5. So really they are just altered versions of our normal cellular proteins. And the unfortunate thing about these altered versions of our normal cellular proteins is that they tend to cause a lot of damage, especially in the nervous system, primarily, at least as far as we know, in the brain. What ends up happening is when we have a healthy neuron with our version 1 of our protein, everything is going well, everything is placed where it's supposed to be, etc. And that comes in contact with version 1.5 of the protein, the altered version of the protein. All kinds of unfortunate events begin to take place. Basically, this altered version of the protein, version 1.5, causes a cascade of events to occur that leads to damaged neurons and therefore damaged brain tissue overall. What these altered proteins do, what these prions do, is cause the normal proteins to kind of tangle up and degrade a little bit. Other structures within the neuron and in the brain will start to fall apart as a result, because normally those proteins are there helping provide structure to the neuron itself and to various membranes, etc. within the neuron. And then once these prions cause the normal proteins to kind of fall apart and get tangled up, unfortunately, it doesn't just end right there in a localized area. It causes all of the proteins that are next to them to do this thing. And so then we have, if this is point A, then point B, which is right next to it, is all tangled up as well, and the neuron starts to degrade. Point C, which would be next to point B, starts with this tangling process, and the neuron starts to degrade. And basically, it's a domino effect. Once one or a few of these altered versions of this protein insert themselves into a healthy neuron or into a healthy brain, like I said, they cause this cascade of events where all of the cells and all of the structures next to them start to fall apart, and then they too become similar to the prions in that they cause damage next to the cells that are next to them. So in addition to the damage that's occurring, the structural damage that's occurring, which is bad enough, this also seems to at least alert the immune system enough to the damage, and the immune system comes and tries to kind of clear away these damaged areas. And in the process of trying to clear away these damaged areas, it sometimes causes additional damage. And unfortunately, the combination of the damage caused by the prion and the damage caused by the immune system is enough to create holes in brain tissue. And as you can imagine, having holes in brain tissue is not a very good thing. People tend to experience fairly rapid cognitive declines. They may have trouble walking, they may have trouble sleeping, they may have memory problems. Exactly what symptoms they have depend on the exact prion they've been infected with. But in general, at this point in time, once someone develops a prion disease or once they get sick, start to show symptoms, there is no cure for this situation. The best we can do is make them comfortable as they go through the process of dying. I usually try to add something positive about every type of microbe. So we know that microbes can be damaging and problematic, but hopefully by now you've learned that they have their upsides as well. And I cannot say that for prions, at least at this point in time. As of right now, prions, as far as we know, are always bad. That information could change at some point. We could someday discover something useful or new about these. But as of right now, prions are always bad, always fatal, and something we hope that no one we care about ever develops.